So thanks for inviting me, and it's a great pleasure to be here this beautiful Saturday. I've got a bunch of pictures and some figures and stuff, so if you do want to move a little more forward during the talk to see better, feel free to do so. I'm going to be talking today about some killer whale genetics and focusing particularly on paternity analysis of the southern resident killer whale population. This is a project that I've been involved in along with my collaborators at NOAA, I don't know, 10 to 12 years, and I look forward to sharing some of those results today. So I'm actually going to go back in time a little bit and kind of start this, not talking about whales, but go back to the 1830s and give a little brief introduction kind of on evolution and genetics so that we're kind of prepared to connect that to what we know about killer whales today. So Charles Darwin was born in England in 1809, so a wealthy country doctor. And his family was a sort of abolitionist family, intellectuals, kind of the sort of liberals of their day. His father really wanted him to be a doctor and follow his footsteps, but Charles preferred to get out into the English countryside and enjoy nature, collect beetles, and so forth, and didn't want to be a doctor. So his father was concerned about this, since he wasn't sure how he was going to make a living, and encouraged him to study to be a parson, so that he could make a living and then do his natural history work as a hobby on the side. So he might have just been a country parson and kind of doodled around the countryside, but for a fortuitous event, his botany teacher in 1831, right after he got his undergraduate degree, suggested that he go along as kind of a gentleman naturalist on a hydrological survey on the HMS Beagle. So he did that. He was not really an official member of the company. He was a volunteer and a guest of the captain. He managed to convince his father to pay his way. And it was during this voyage that he started to gather some of the ideas that ultimately, information and ideas that ultimately led him to propose his theory of evolution and natural selection. So when he returned to England in 1836, he started kind of writing some of these things down, where he drew what might be kind of the first hypothesized phylogenetic tree, illustrating the idea that species exist in a kind of ancestor-descendant relationship. So the sort of diversity of life that we see today shares a common ancestor sometime in the past, and has evolved in this kind of tree-like fashion. So he started writing about that, and at the same time, around the same time, a year later, he ended up marrying his first cousin, Wedgwood, in 1939. They had a very successful marriage, 10 children, and he then spent the next 20 years working on his theory of evolution and natural selection, along with publishing some books on barnacles and coral atolls, and of course the voyage of the Beagle. Then ultimately, in 1859, he published his seminal book on the origin of the species, where he promulgated these ideas. This was an instant bestseller, and it's become probably one of maybe the most important biological idea ever. One problem that Darwin had, though, in all of this, was that he had at best a very fuzzy idea about genetics, and he realized this was a problem for his theory. The kind of idea of how genetics worked at the time was one of blending inheritance. Basically, the assumption was that an offspring was sort of a mixture of its mother and father, somewhere in the average of the two, and this didn't seem to work very well with the idea of evolution by natural selection because it wasn't really clear where variation came from and what would happen to that variation even if it arrived. So this was always kind of a weak spot of his theory. The theory of genetics that he published along with the origin of the species, was entirely incorrect. So this leads me to a contemporary of Darwin's, Gregor Mendel, born about 10 years later in what's now the Czech Republic. He was born into a poor family, a farming family, not terribly wealthy. He was a bright boy and went to the local high school. And actually had to use his sister's dowry to pay for that. But he did well there. And his father, kind of like Darwin's father, suggested that he go into religious order in order to pay for his studies. So in the mid-1800s, he became a priest, or started to become a priest, at the St. Thomas Abbey in the town of Bern. And again, it's down in the Czech Republic. There's a picture of it here. I've never been here. This is just pulled off the web, but here's the abbey as it still exists today. And here's a picture of where he started to do his experiments on peas. So Mendel's an interesting person. He actually was studying to be a high school teacher. That's what his goal was. He ended up twice failing to get his teaching certificate. And so kind of giving up on that, he decided to focus on experimental biology and become the founder of modern genetics. So Mendel did his work on peas, and he conducted this work over a period of about 7 years, from 1856 to 1863. And his work is important in a number of ways. One, it was one of the first really large-scale experimental studies where someone sort of developed a very clear hypothesis, conducted a lot of experimental work, conducted statistical analyses to test those hypotheses, and then kind of modified it and moved forward from there. He looked at a total of 28,000 plants over this 6 years, and then produced a really important paper at the end of it. So he, in his experiments, found two very important discoveries on how information is transmitted across generations. The first was that instead of the kind of blending inheritance idea that Darwin was thinking of, he figured out that inheritance is particular, that there's particular things that must be moving between the generations, and that those things don't really change. They're passed unchanged from parent to offspring. And so this graph here, I don't know if you see I don't know if you can see this. But anyway, so on the so here's an example of this with these pea plants. And in this case, he took a tall plant and a short plant and bred these plants together. All the plants are tall. They're not intermediate or kind of medium-sized plants. So in this case, not really blending inheritance. But then when you cross these tall plants together in another generation, you recover some of these short plants. And so he was able to kind of infer from this that there's some aspect of shortness that is transmitted across these generations in a complete and whole manner and can manifest itself again in subsequent generations. So this idea of this kind of particulate nature of inheritance is really important. And based on the numbers and patterns of this, he was able to determine that you've got one particle from your mom and one particle from your dad. And in this case, if you have two short particles, it would be short. If you have two tall particles, it would be tall. And if you have one of each, it would also be tall. So that leads to kind of the second kind of important aspect of genetics that we discovered. That's the idea of dominance and recessiveness of genes. So in this case, the short particle, the short gene is recessive to the tall gene. It's only expressed if both copies of that short gene come together in the same plant. So Mendel looked at not just short and tall, but a whole bunch of different traits, and all of which kind of seem to behave more or less this way, and came to these important conclusions about how genetics works. He published them I talked about them twice. He gave 2 presentations of his work in 1865 at the Society of Natural History of Peru. And he published 1 paper in 1866 in the Proceedings of that society. This was not a compact journal. These were published in a kind of relatively obscure way, and hardly anyone read them. His work was cited twice in the next 35 years. And Darwin, most importantly, never heard of Mendel and never read any of his work. Mendel's work was rediscovered 35 years, 34 years later, around 1900, when 2 other geneticists, both botanists, independently basically did the same sorts of experiments and found the same thing. And as they were writing up their papers, they did a literature search and found Mendel's earlier work and realized that he had done it first. And so his work was rediscovered. He was long dead. He died in the 1880s, so he never lived to see his work really acknowledged. But he's now regarded as the father of modern genetics. So from 1900 to about 1950, the sort of main kind of focus of genetics was trying to figure out, well, what these particles are. What is a gene? Mendel's work showed that something is being transmitted across generations, and the question is what it was. And it turns out that genes are chemical, and the chemical is deoxyribonucleic acid, or DNA. They're spooled up into chromosomes, and just as Mendel inferred, your typical diploid organism gets 1 set of chromosomes from their mom, 1 set of chromosomes from their dad, and all of the genetic material that is necessary to create an organism and allow its development is stored in the sequence of bases in this DNA code. So there's 4 letters in the code, G, A, C, and T, that stand for 4 different chemical subunits. And your typical mammal contains about 3 billion base pairs, 3 billion letters in their genetic code that code for about 25,000 genes. So that's true of humans. It's also true of killer whales. So this was kind of discovered in the 40s and 50s. The structure and function of DNA was discovered in the late 50s. And then kind of since then, it's been a race to really kind of figure out how everything works. 1972, the first gene was sequenced. In 2003, the first whole human genome was sequenced, only 2003. And in 2015, just 2 years ago, the first killer whale genome was sequenced. So, you wonder when I can start talking about whales. So, it's starting to kind of connect this a little bit with the southern resident population. Right around the same time period as Darwin was formulating his theory and Mendel was working on genetics, the sort of antecedents of the southern residents' decline, if you will, had started here where we live in the form of European settlement of the Pacific Northwest. So here, for example, is what Seattle looked like around 1870. So in the same time period, these scientific discoveries were made. And as I think many of you know, the southern residents focus on eating Chinook salmon. In 1866, the same year that Mendel published his important paper, the first salmon hatchery was built on the lower Columbia River at the Eagle Cliff area. So kind of right around the same time period, the sort of factors that lead us to be concerned about the conservation of this population were starting to manifest themselves. So what, to me, it's kind of an interesting connection to kind of think about the sort of origins of this science is kind of happening around the same time as the city of Seattle and its areas that live in town were So, as you mentioned, Darwin drew this first hypothesized phylogenetic tree around 1837. I just thought it'd be interesting to connect this to the whales and what our phylogenetic trees look like now. This is kind of a current view of a phylogenetic tree of all of life based on DNA sequences. So you take the genomes of lots and lots of organisms and line them all up and use that information to draw a phylogenetic tree. Killer whales and humans are kind of right down here on this tree. In fact, all eukaryotic organisms are here. Everything else on here is mostly bacteria. So most of the kind of diversity of life, at least at the DNA sequence level, occurs among not the big organisms that you see around you, but the tiny invisible organisms that you don't see. So let's start kind of focusing in a little bit on killer whales. This is a figure from a paper by Andy Foote et al. It was just published a couple of years ago. That actually was the first paper on the killer whale genome. They published the entire nearly 3 billion base pairs of the killer whale genetic code. This was not really the topic of the paper, but it's got a nice figure that illustrates that killer whales, along with all other cetaceans, evolved from land animals. They were not originally aquatic animals, but evolved from ruminants, like related to cows and sheep of today. Interestingly enough, Darwin actually had a hard time with whales, and a lot of people challenged him to explain how a whale could evolve. And he hypothesized in the first edition of the Origin of the Species that maybe a bear started swimming in the water and maybe eating fish, and over time slowly developed a blowhole and the morphology of a whale. So he kind of had the right idea of the wrong organism. If he had been talking about a walrus, he would have been right on track. Those did evolve from some of the bears. Focusing in on just cetaceans now, here's another phylogenetic tree, again based on DNA sequences, showing most of the current species of cetacean. Some examples on the right. And the only point here is that killer whales are delphinid, related to dolphins. They probably split from other dolphins about 10 million years ago based on these data, and from other whales, other marine whales, about 40 to 40 million years ago. Focusing in even a little more closely on killer whales and diversity within killer whales. Killer whales currently are considered a single species that is distributed all around the world. There's a lot of variation in this species, a lot of both morphological variation and ecological variation. So here's a figure from a paper by Reich et al. A couple years ago, kind of describing this. So we've heard of these variants of killer whales as ecotypes, and these are groups of killer whales that specialize either on a particular prey or it might display a particular behavior or occupy a particular habitat. There's a lot of work ongoing right now on trying to understand the genetic relationships among these different ecotypes and when they evolved, and whether some of these ecotypes might in fact be distinct enough to be considered species in their own right or subspecies. I don't really talk about all that today, other than to note that there's a lot of work, a lot of it is in progress and even contradictory at times. But some of the things that I think we've learned from the genetic studies of these different ecotypes are, one, they're all relatively recently evolved. So most of these ecotypes have formed and evolved within the last couple 100,000 years. Actually, roughly around the same time period that humans have sort of evolved into a global population. The killer whale ecotypes seem to have done that on roughly the same time frame. But they are genetically coherent, and generally the different ecotypes you can tell them apart genetically. They're somewhat genetically distinct. The way that these ecotypes have formed may be related to kind of the matrilineal structure of a lot of these populations, where A small population might go through a population bottleneck, and by going through that kind of small population size, it's an opportunity for the population to kind of change ecologically and behaviorally in a way that makes it different from other populations.
I'm having trouble hearing you.
Oh, I'm sorry.
Thank you.
How's this?
Way better.
Okay, my apologies, sorry. It's hard for me to tell what the sound is like out there. So here in the Pacific Northwest, we have three ecotypes, residents, transients, and offshores. And so residents are a fish-eating killer whale. They have a very stable matrilineal social structure. They're very highly vocal. They're called residents, but it's a little bit of a misnomer in that they actually range quite widely. The southern resident population that I'll be talking about today occupies an area all the way from California up to occasionally southeast Alaska. So big range, and particularly in recent years, portions of the population probably spend more time on the coast of Washington and Oregon than they do in this area. The residents, as I mentioned, are entirely fish-eating. If you look at their diet, they do occasionally play with other marine mammals. So this is a harbor porpoise, and kind of I think the killer whale here is kind of playing with the porpoise the way a cat would play with a mouse. It also kind of illustrates these are two relatively closely related organisms, and evolved, again, probably about 10 million years divergence, and obviously very different in size and behavior, but somewhat similar in shape and morphology. The other commonly observed ecotype around here are the transients, or Biggs killer whale. These are entirely marine mammal eating. So here's a predation event on a sea lion. So he's not playing with the sea lion. And then the third ecotype that you don't see very often in inland waters, but is more common on the outer coast, is this offshore group, which also appears to be fish-eating and is genetically related to the resident whales. All right, so coming in even kind of now closer, just focusing on the resident ecotype. The southern resident population is just one of probably half a dozen of these fish-eating killer whale populations that inhabit the southern resident population is down here. The southern residents are the most southerly distributed of this group, and they're also somewhat anomalous in that they're one of the only groups that is sort of not recovered well after being protected by the Marine Mammal Protection Act in the mid-1970s. So for the most part, killer whale populations are actually doing pretty well, and have been increasing. There are a couple of exceptions. The southern residents being one of them. So focusing on even further, so we've kind of gone from the tree of life and everything down to cetaceans, down to delphinids and killer whales and ecotypes and the resident ecotype. We can go even further and talk about acoustic clans. Those are within in populations, and those are groups of killer whales that have kind of the same dialect or the same sounds, and then pods, which are kind of groups of related killer whales. So for the southern resident population, we have JK and L pod, and then macrolines, which I'll be kind of focusing on for the rest of the talk, which are females and her female and her immediate descendants. So as Susan mentioned, the southern resident killer whales were listed as endangered under the Endangered Species Act in 2005. The main risk factors that cause them to be at risk are, one, simply a lack of population growth and a small population size. So simply their demography is such that they are at risk from just small population size effects. And then potentially lack of prey quantity and quality, contaminants, and then disturbance from boats and noise. So our research program at the Northwest Fisheries Science Center focuses on all of these. Today I'm really only going to be talking about the breeding structure of the population, and that's relevant to this risk associated with abundance and demography. So how does breeding structure affect risk? There's a couple of ways. One, genetic diversity is sort of learned from Darwin, is important for allowing populations to adapt and respond to environmental changes. Diverse populations have what they need to do that. And the breeding structure affects how much diversity there is in a population. Both how much, if you have interbreeding with other populations, that can increase diversity. If you don't, it reduces diversity. And then even if you don't have interbreeding with other populations, kind of how the breeding structure occurs within a population affects how much diversity is in that population. So for example, the number of breeders, if you have most of the population breeding with kind of an even variance in breeding success, that tends to promote genetic diversity. If you have only a couple breeders, that tends to reduce diversity. And then whether relatives are breeding. So if you have inbreeding, inbreeding tends to reduce genetic diversity compared to non-inbreeding populations. And then finally, the breeding structure, some aspects of the breeding structure can simply affect actually maybe even just the population growth. So for example, if there are certain customs or taboos or something that limit breeding, say on pods or something, that could potentially actually limit population growth due to a lack of opportunity for appropriate mates. So all these things are kind of hypothetical in ways that the breeding structure might affect extinction risk. So why do we use genetics to study this? And there's a couple of reasons. One is that there's often a really big difference between the kind of social breeding structure that you might observe simply by watching a population and what you learn when you actually look at the genes or see kind of really who's breeding with whom. And a great example of this is with birds. So in 1970, the kind of state-of-the-art general feeling amongst people studying passerine birds was that they were nearly all monogamous. And that's because when you go out and look with a pair of binoculars and kind of watch who's mating with whom and who's nesting with whom, it sure looks like they're monogamous. But over this time period when DNA sequencing became available and scientists could actually study the transmission of genes in these birds and figure out who the dads were, it turns out that actually they're not monogamous at all. And most passerine birds are in fact polygamous. And the kind of social dad who's on the nest is not the only dad of the chicks in that nest. And the same is true for marine mammals. So for example, in the seals, again, you can go out with a pair of binoculars and kind of watch who's living with whom, and you might kind of get the idea that the big dominant male is the only guy doing the breeding. But it turns out when you go into the genetics, a lot of the pups are sired by more kind of junior males, if you will, and those matings are occurring in the water where you can't see them. And so you wouldn't know that if you just watch the population and do the genetics. And then finally, a lot of things like whales or other animals that are difficult to study, it's usually easier to get a genetic sample than it is to observe the animal in some cases. So if they're in an aquatic habitat, for example, or if they're simply not observable for much of the time, it might be easier to do this with genetics than observation. So for studying killer whale opportunity, the first thing we have to do, well actually, let me back up slightly. In the case of killer whales, the maternal relationships, so basically who the moms are, is well known through decades of study. So starting in the early 1970s, Canadian researchers like Michael Bigg, and then later John Ford and colleagues would go out and watch the whales, and by observing the whales, you could pretty much, and then up here in this area, Ken Malcolm and Center for Whale Research started this research in the mid-70s. And simply by observing the whales, it's fairly readily apparent who the moms are. A calf shows up, she's with her mom, he's with her mom, and so the maternal relationships and who the moms are relatively well documented. The question is who are the dads, and that you really can't figure out just by watching the whales. So to figure that out, the first thing you have to do is start to get tissue samples or some way to collect DNA from the animals in the population. And we do this in two different ways. The first and kind of most effective and important way is to collect a biopsy sample. So here's Brad Hansen, who's the head of the marine mammal team at the Northwest Fisheries Science Center. And he's got a pneumatic dart gun here and is collecting about an inch long sample from a whale, about the diameter of a pencil. And this is our most important source of DNA from these whales. And the reason why this is a great source of DNA is twofold. One, because you're getting a little bit of the skin, you get a very high quality sample, so the DNA is easy to use and easy to study, and you get a fair bit of it. And then two, kind of most importantly, because you're right next to the whale, when you take that sample, you know exactly what it is you're sampling. There's no ambiguity that this sample came from this particular whale. Our other source is also an important source, but it's maybe slightly more difficult to use, and that's fecal samples. So there's a lot of DNA in poop, and we use these samples for all kinds of things, particularly for studying the diet of the whales, where it's invaluable. But we can also get whale DNA out of the poop samples. The sort of cells from the intestinal tract end up in the poop, and you can get whale DNA from those. So these are collected basically by following the whales around in a small boat with a swimming pool net and simply scooping the poop. These are great samples. We collect a lot of them this way. We also collaborate with Sam Wasser at the University of Washington, and he and his students use dogs who have been trained to smell out whale poop and collect it on the water. So we get a lot of samples like this, many more poop samples than biopsy samples. The one kind of weakness of these samples is that we typically don't know exactly who did the pooping, particularly with the dog samples, which are not collected near the whales. So these are kind of an important secondary source of samples. At this point, we actually have most of the population biopsy sample. So how do you do a paternity analysis? It's really just like, it's kind of using the same principles that Mendel used and discovered in his peas. So, as I mentioned, there's about 3 billion base pairs in the genetic code of a killer whale. We go in and look at, for this particular study, we're looking at about 90 of those 3 billion sites. But that's enough information for us to figure out who the dads are. And so the principle of that is illustrated here. So we have a non-killer whale, and we have a group of several dads who are the possible dads of this mom's 2 calves. And so in this case, we go in and we sequence the mom, and at this particular place in these 3 billion base pairs, we discover that this mom received a T allele from her dad, and she also received a T allele from her mom. So she's a TT. We go in and look at her calves, and one of her calves is a CT, and one of her calves is an AT. So we know that T had to have come from mom, because she's just a TT. So in this case, the A and the C of these 2 different calves had to have come from the dad. With me so far? So then we go and look at these dads. So we got one dad who's a TT, another one who's an AA, and another one who's a CA. So from this, if we're confident that these 3 are the only dads who are the only possible dads, and if we kind of rule out things like mutation or other things that might screw things up, we can say with some confidence that the C dad has to be the father of the calf that has the C allele. With the other calf, it's a little more complicated because we have 2 dads who have A's. We have a CA and an AA. And so either one of those dads could potentially be the father of this calf over here and contributed that A allele. And, you know, we might count things up and say that, well, the dad who has 2 A's is maybe more likely to be the father than the dad who just has 1 A's, and that's true. And so we might say he's a little more likely, but we can't rule it out entirely. So this is the principle we use for paternity analysis. Instead of looking at just one site, we look at a whole bunch, which gives us a lot more power to do that. But that's the basic idea. So let's look at a real world example of this. So this is female K14 on the left and her calf K42 on the right. K14 is one of the older females, born in the mid-70s, and her calf was born in 2008. And the question is, who is dad? And drum roll. There we go. So here are all of the potential dads. So because this one was born in 2008, relatively recently, we actually have samples from, there's basically 20 dads, roughly about 20 dads in the population, 20 males, sorry, in the population who could be this calf's dad. And here they are. And we have samples from basically all but one of them. There's one dad who's not here, but we have samples from everybody else. So nearly all the dads are genetically sampled. And you can see the way we do this is we kind of give every male the benefit of the doubt. So we assume that any male over the age of around 10 or 11 is at least a potential dad. And so some of these look pretty different. So for example, you know, this picture here, we have L41, and then L88 and L89. L41 is an older male, very mature. These were not, these two at the time of this picture were back. Sorry, I should not use the laser pointer if I accidentally move things. These files are big enough that you need to go slow on hitting the buttons there. Okay, here we go. Anyway, so in that bottom picture, you have like L41, who's a very large, very mature male, and some younger males who are included in the group of potential males, but are less mature and probably less likely to be the dads, so we give them the benefit of the doubt. So in this case, we go through our analysis, and it turns out it is. In this case, L41 was the dad of K42, and there's no ambiguity in this particular result. There's really no other dad that could be. Turns out he's also the father of K42's older brother, K36. So those two share the same dad, are full siblings. But he's not the dad of their other sibling, K26, who has some other dad. And in fact, K26's dad is not among the ones that we have sampled, so he remains an unknown father. Probably because K26 is an older whale, and his father presumably died before we had an opportunity to sample. In some cases, so for most of the assignments that we do, we know who the mom is. And we're just trying to figure out the dad. But we sometimes end up in situations where we don't know who the mom is. These are often kind of sad situations, but important nonetheless. So amongst our samples, we have 3 or 4 cases where we have a neonate calf or some young, very young animal who is washed up on the beach somewhere before it was ever observed in the population. So you don't know who the mom is. And we're interested in knowing both who the mom and the dad is. So here, for example, this calf, this neonate calf, washed up on Dungeness Spit in 2013, and we had the opportunity to try to figure out who the parents were. In this case, we can show that it was the mom was J28, and the dad was also L41. So, what does this look like if you kind of put it all together? And this is where you might want to sit in front. It's a little hard to see. That's okay, because let's deal with the colors here. So this is a family tree of the whole population that shows every individual in the population who has ever been observed is a dot on this tree. So each of these little dots that you see are an individual whale, some of whom are dead, but it kind of shows the entire population. And they're ordered from bottom to top in their birthdays. So the older whales are at the bottom, the younger whales are at the top, going from the early 1900s up to the present. These older dates are kind of guesses. Each mom and calf is connected by a line, which I acknowledge is a little bit hard to see on the screen. And so all the natural lines you can see are kind of along the screen here. The dads are color coded, so each of the dads is a triangle. So here's one dad. This is J1. Here's another dad. This is L41. And there's a total of 8 males that have been identified as fathers in the population. So J1, J26, K26, L41, L57, L78, L79, and L85. There's a couple things to kind of notice here. One is that, then each dad is connected to his offspring by the color. So everything that's red in here are offspring of J1. Everything that's blue, those are the 2 dominant ones, are the offspring of L41. And the others are color-coded accordingly. So there's a couple things to notice. One, we have a lot more paternities in the more recent data. And that's simply because we have those fathers sampled. So we have no idea who the dads are of these older animals who were born a long time ago, because their dads have been lost to history and never were part of our sample. So most of our information is based on younger animals. And then the second kind of obvious thing from this is that the variance in breeding success is pretty extreme. There's basically two animals here who are responsible for 85% of the paternity assignments, and those are J1 and L41. So what do we know about these guys? So you have J1 on the top, L41 on the bottom. J1 has been the oldest male in the population, was the oldest male in the population throughout the entire study period. He was a mature male.